We have Dr. Eduardo Amorim with us today from Cal State Northridge. And Eduardo actually did a postdoc here at UCLA not too long ago. He was sharing that it's nice to be back on campus. And he's been very busy since the time that he was here. Doing all sorts of really cool things with dog genomes, ancient genomes of humans, human adaptation to altitude. And today he's going to share with us his current work that focuses on human immune evolution. And so please welcome. Please welcome with me Eduardo today.
Thank you so much. Can you hear me on the camera? Okay. Yeah. Thank you everyone for coming, and thanks, Abby, for the invitation and for the introduction. Today I'm going to be talking about some of the work that I'm doing in my lab related to using ancient DNA to study human evolution in response to pathogens. And in my trajectory as a postdoc was actually pretty long, typically for postdoc in the United States. I worked with 4 different people, and I worked with 2 people that were focusing on population genetics, and 2 people that were focusing on paleogenomics or ancient DNA studies. And with the people that I worked with population genetics, I was mostly studying natural selection, deleterious variation, but also adaptation to environmental pressures. And with the people that I worked with with paleogenomics, I was working with history reconstruction. And actually in 2019, I gave a talk here at Beck also on my work about the barbarian migrations using ancient DNA. I don't know if anyone was here at the time. It's 5 years ago now. And so those are the 2 things that I learned about during my postdoctoral training. Those 2 things are my passion, and I always wanted to put those 2 things together. And I was able to do it towards the end of my postdoctoral training. And now, more specifically, I've been able to put those 2 things together in my own lab. So my idea in my lab is that I want to leverage the information that we have from paleogenomic data sets that have information about several different time points in the history of humans, for example, and use that to learn about adaptation. And I'm doing that now with a team of many different collaborators. I still work with Ana Malaspinas, who was my last postdoctoral advisor. Lucas, one of her students. My lab at CSUN here in Northridge doesn't have an ancient DNA lab. I don't have access to that in my institution, so I use Lars's lab at Santa Cruz. So I have to thank him for a lot of the stuff that I'm going to be discussing here today. Thanks to him, this is happening. Part of my work is happening. And in my lab, there are 2 people currently working on this project. One is Flavio De Angelis, my postdoc, and the other one is my student, Caroline Solis. And we also got a grant from the NIH to study human host pathogen coevolution, and also pathogen-driven natural selection. So I also want to acknowledge that. So this is perfect. I managed to put those 2 of my passions together, and now I am in my lab doing this type of research. I'm going to talk to you a little bit about one of these projects, but I want to show here the types of questions that I ask in this type of framework. One is about pathogen-driven natural selection and host pathogen coevolution, which is going to be the focus of today's topic. Another thing that I study is human adaptation to hypoxia, specifically in humans living in the Andes in South America. And the 3rd topic that I study is a bunch of different things related to dogs, domestic dogs, wolves, and other species of canids. I'm interested in the relationship, in the interaction between dogs and canids. For example, domestication, but also extinction caused by humans. So those are the 3 different topics that I study in my lab currently. And today I'm going to be talking about the first one, as I mentioned. Excuse me. And if we have time, also I have some data to discuss during the discussion about the second topic, which I think would be of interest of other people here. It's still very early on, so I just have some data and some ideas and some questions. But I would love to talk more about the second project with you too, later in the discussion. Okay, so before talking about specifically about my project using ancient DNA to study pathogen-driven natural selection, I want to talk about give you a big picture of what's going on in the ancient DNA world. What are the populations, what are the time periods that are being studied, and how my lab fits into this. So as of last month, there were data for almost 10,000 human individuals, ancient or historical humans, that are publicly available online. It's a pretty large number. This paper from 2021 shows you how many samples were generated, data for how many individuals were generated each year. So we see that this is increasing, and now we have 10,000 individuals that we can have data for. And you see here, I'm going to come back to this later, you see here that most of the samples are concentrated in this region here, right? So Western Eurasia. And there are some samples coming from different sites in the Americas and also from other countries of the world, but mostly from Europe. This is still the picture nowadays, but I don't have a map showing that, but this is still pretty much what we see today. The dates that people usually study with ancient DNA varies a lot. There is no specific date that, at least to my knowledge, that we start calling, oh, this is ancient DNA. Basically, we can study things that are like a few decades ago, up to over 30,000, 50,000 years ago, and so on. And with this data, a lot of you might know already that in 2022, Svante Pääbo, a researcher in Max Planck Institute in Germany, won this Nobel Prize for his discoveries concerning the genomes of extinct hominids and human evolution. That was a very big surprise for us. Big surprise, but also like very positive surprise, right? For us in the field of ancient DNA, paleogenomics, that our science was recognized for a prize of this caliber. And the questions that his group are focused is mostly related to archaic and modern human interactions. Archaic, for example, the Neanderthals, and modern humans is us. So they are studying mostly the history of archaic humans and the interactions that they had with us. But those are not the only types of questions that we can ask with ancient DNA. If you were here in 2019 when I was talking about the barbarian migrations, I was talking about human population dynamics and movement, migration, and also about kinship inference. So those are two things that we can also do with ancient DNA. We also can recover the lost genetic lineages. For example, recently one of our projects, we were able to recover a 7,000 years old Treponema strain in an individual from South America. And this strain has completely disappeared nowadays. The strain that we have in South America currently was brought by Europeans, and we were able to discover this new strain that wasn't described before. So this is also one of the advantages of doing ancient DNA, and one of the interesting things that we can do. And last is the characterization of ancient human pathogens, which I just mentioned. Just so you know, ancient DNA can be obtained from human samples from tooth and pieces of bones, of course, but we can also sometimes recover pathogens that were circulating in the blood of that individual, especially when we take DNA from the blood. So we have this chance to also characterize ancient pathogens that were infecting humans in the past. This is pretty much the core of what paleogenomics is today. Those are the different topics that different groups are studying. And more recently, actually in 2015, we have one paper. I'm going to show some results later. And then later, a couple of papers focusing on phenotypic reconstruction, okay, like trying to see what's the pigmentation phenotypes or maybe height of ancient individuals based on genetics. And more recently, people started to look into natural selection as well. And you are probably familiar with the term survival of the fittest from Charles Darwin. And that's one of the initial ideas that the community, scientific community had about natural selection. I like this definition here from my colleague Ziwe Gao. She says, selection favors genetic variants that lead to advantageous phenotypic changes in specific environments, resulting in increases in allele frequencies over time and distinctive pattern of genetic variation in present-day populations. The part that I want to highlight here is this increases in allele frequencies over time. Allele is just one form of a gene, for example. So we have like a mutation bring a new form of that allele, and we have, for example, the ABO gene, for example, the blood type. We have allele A, allele B, and allele O. So they're just forms of the gene. So what natural selection does is when there are two variants, these variants might change in frequency for like random factors, but also because of natural selection. And if one of those variants is advantageous, the frequency of this variant is going to increase in time. So this is the effect of natural selection that we, in theory, could observe if we had ancient DNA data to do that, right? But what we usually do is only looking at the present and try to infer. Most people do that, let's say, looking at the present day variation, trying to infer what happened in the past. But let's look into the past first. So this is an allele frequency trajectory. I have on the y-axis the allele frequency. It's coming from nearly 0 to nearly 100%, let's say. And then the time goes from the left to the right. So we see that there is an increase in allele frequency over time. And what we can see here is that the selected variant, for example, the advantageous variant in a gene, it starts to be more frequent in the population. So we see here those little red dots showing that, and here in the past we had only 2. And before ancient DNA, before we were able to actually see this trajectory over time with ancient DNA, we would have to use what Ziwe is saying there in her that sentence, distinctive patterns of genetic variation in present-day populations. If you see her review came last month or this month, it's pretty interesting. She shows many different types of methods. I'm showing here just the 3 that for me are the most popular ones. One is based on the what we call the side frequency spectrum, which summarizes the allele frequencies of different genes across the genome. The other one is a very popular one, which is this allele frequency differentiation. That means each population that we study, like the 3 sets of populations there, they have a certain divergence between themselves, right? One is more divergent than the other 2, and the other 2 are more closely related, let's say. We can use this information to look for genes or for variants that are actually outliers in relation to this tree. So genes that show a larger divergence than the average of the genome, for example. This is another method. And last, there is this haplotype structure. It also changes, natural selection will also change the extent of haplotype homozygosity in the genome. So those are some methods. Those are always looking at the present and trying to leverage this information, or let's say, leverage information from the present to understand this, phenomena that happened in the past. And with these types of papers that many people are studying, probably there is a lot of papers from Abi here in this review. A lot of us in this field has contributed to this type of figure here, showing that different populations around the world adapted to their specific environmental conditions. And here we see a few different themes here. We see a lot of as examples related to diet, right? High-fat diet, lactase persistence. We see traits related to diseases, for example, malaria, trypanosome resistance. And also we see adaptations related to extreme environmental conditions, such as toxic arsenic-rich environment, Arctic environment, and high altitude. So those are a few themes that we can study with this type of framework, looking at the present to see what happened in the past in terms of local adaptations. And my idea, as I mentioned before, I wanted to put the population genetics that I learned with present-day data, together with the ancient DNA data that I used for history reconstruction, putting those two things together to learn about natural selection. And so here are the two questions that motivated this. One is, can we use ancient DNA to learn about these allele frequency trajectories and thus infer natural selection? And what are the novel information that we can learn from ancient DNA that we can't just using present-day data? So here's some simulations that we did for different allele frequency trajectories. The population here has specific size that doesn't change across time, and this population evolves through 100 generations, from 0 to 100 generations. And what we can see here is a bunch of different trajectories. Here on the y-axis, we see the allele frequency, and we see that they always start at 10%, and sometimes it reaches 100% in the case of this red curve here. And those curves, they represent different selection coefficients, basically the strength of selection. We have 0.2, which is a very strong selective coefficient, and very weak, like 0.001, and neutral in green here. And we see that depending on the selection coefficient, this allele trajectory will be different. So we're doing some work trying to understand what's the best sampling strategy to follow, to implement in order to learn about this sampling. When, for example, selection is strong, like the red curve, it's better to have things that are more spaced. Whereas when you have, for example, things that are selection that it's weaker, it's better to have a longer generation time that you observe. Because otherwise, if you look just like in a short time period, for example, you don't observe the differences between these selection coefficients. So we're trying to understand this better to see what type of data actually do we need to build with ancient DNA to actually be able to study natural selection. And we're using some simulations. But here you can see that basically everything else being the same, just the change in the selection coefficient, we see that there is different allele trajectories, right? So the shape and the slope of this trajectory tells us about the selection coefficient, which is something that typically tests using present-day populations and no experimental evolution studies, just like looking at present-day polymorphisms. You can't actually infer that. And the other thing is that ancient DNA can also provide us with an estimate of the time of onset of selection. So we can see if the allele frequencies are varying like they are neutral, and at a certain point they start to increase in frequency. And we can infer that around that time, selection happened. So those are the two, this is the idea of using ancient DNA, right? And those two are the two advantages of using ancient DNA over just present-day data.
Can I ask a question
sure.
of information? Qualitatively, what you've said is totally clear, but I was just wondering if you could tell us how the selection coefficient is calculated, or is that too complicated?
The selection coefficient, there are at least two ways to calculate selection coefficient, but basically when we're doing simulations, we measure the proportion of individuals that will survive. Let me see. The selection coefficient will be related to the probability of that individual passing their allele on to the next generation. So it's about proportion of chances of passing your genetic heritage on to the next generation. So it reflects that. We can talk later more about some details, but basically it reflects this your probability, relative probability of you transmitting your genes to the next generation. And then there is also some complications about being dominant or recessive or being additive. There are different types of inheritance. But basically, this selection coefficient will tell you if you have a higher or lower chance or a neutral chance to pass on your genes. So this all sounds super cool, super exciting. But we are actually struggling a lot to implement these ideas because studying ancient DNA comes with a lot of challenges and a lot of limitations. I'm not sure if anyone here is familiar with working with ancient DNA. I'm going to go over very quickly this, but we can discuss more later because I could spend 1 hour talking about this. But those are all some problems that we deal with. And we also always have ways to correct that. One of the problems is that ancient DNA is degraded. So when the individual dies, DNA is there and it starts to interact with water, oxygen, and other factors, and it starts to degrade. It starts to be fragmented. It starts to accumulate mutations that were not there when the individual was alive. Basically, it happens also when we are alive, but our repair mechanism corrects that. And when we are dead, that doesn't happen anymore. So these mutations start to accumulate. And that's what we call post-mortem damage. This sounds terrible, right? But at the same time, this is actually very assuring when we see that in a sample because we know, oh, this is actually ancient DNA. It's not something else. And then we can treat that. We can eliminate those mutations, for example. We can deal with that. That actually sounds bad, but it's actually something that we use in our favor. The other thing is that there is low endogenous DNA content because of this degradation of DNA. Our samples we spend a lot of money sequencing, and maybe 1% of that is human, and the rest is just like soil, bacteria, organisms in the soil, you know. The other thing is low coverage. Typically, I would say that we have 1x coverage, meaning each position in the genome, we only have that sequence once. And that makes our data to have an excess of homozygotes. Because if the site is a true heterozygote, so you have two alleles, right? One allele A and allele B, and you only sequence one of them, it's gonna look like it's a homozygote. So there is all these issues that we have to deal with. And contamination risk, of course, but that's why we have specific clean labs that we call to do ancient DNA analysis. And the last thing here is biases in sample representation, which is something that it's actually not only relevant for paleogenomics, but for human genomics in general, is that there is always an overrepresentation of Europeans in genomic data sets. And there are several reasons for that. In human genomics, we are changing that, right? The NIH, the NIGRI is actually focusing on understudied populations now. But in ancient DNA, we also have that. We also have this overrepresentation of Europeans. And in addition to that, we have this underrepresentation of certain periods in history. And if you can see here in Europe, for example, we have this blue pie, which is less than 2,500 years before present, meaning the historical period, roughly. We see almost no samples there in Europe from that period. This is 2016. Things changed since then, but there are still underrepresentations of other people than non-European and of certain periods. So my lab is working towards addressing those two issues. And today I'm going to talk about some data some study that we're doing in Europeans. The idea is that we're going to expand this to also include Native Americans from South America, where we are already studying. But right now I'm going to focus on the Europeans. And so my lab is working towards those two, addressing those two gaps, studying periods that are understudied and studying populations that are understudied as well. And also developing methods to study ancient DNA, natural selection using ancient DNA. And again, I want to thank those two people, Flavio De Angelis, who is the author of the data that I'm going to be showing today. He's my postdoc, and he is the person behind the data that I'm going to be showing today, and also the person behind generating the data at Lars's lab. And down here, I want to also acknowledge the participation of several different people, students at UCSC and in the University of Lausanne, and my students that are contributing to different pieces of this work. This is what we are currently studying from Europe. Flavio comes from Italy, and he has a lot of contacts there, so we were able to actually get a very good sample, time series data set from different regions of Europe representing different time points. We are looking forward to use this data to study natural selection, but also we are studying changes in the oral microbiome that we can also get from getting dental calculus, for example. So there's a bunch of different questions that we are studying, focusing on Italy specifically. We have samples from before the Roman Empire, after the Roman Empire, so we can see some transformations, how they impacted diet, but also how they impacted genetic variation. And we are looking forward to working with this data. We have currently data for about 300 individuals coming from those sites. And my specific focus, my biggest interest is the historical period. And that's because, including the migration period and the Middle Ages, right? Because there were several disease outbreaks in that time, and also several events that caused important social transformations, like the fall of the Roman Empire, the barbarian migrations, for example. The origin of many current European nations also is dated to this period. So I'm very interested to study this period that is also understudied from the paleogenomic perspective. In South America, we have currently data for 70 individuals approximately coming from different sites, mostly from Peru. And we have about 2/3 of these individuals coming from high altitude, and the rest coming from coastal or from the Amazon. Our time series for South America is not as good as we usually have for Europe. We still have a lot of gaps. Here's an example of a data set that we have with populations from the high altitude. We have a lot of individuals from present day, about 50-something. That is public data, actually. It's not ours, but we are generating data for those ancient ones. And you can see here that most of them come from the period between 500 years ago, just before the contact with Europeans, up to 1,000. And very few actually come from older periods. So our lab is now trying to address this gap for us to understand all the processes that happen related to evolution and history, like adaptation to high altitude and so on, in South America. And the other topic, in addition to generating new data, as I mentioned before, my lab is also developing new approaches to study natural selection using ancient DNA. And this is the work that I mentioned before, that it's done by Flavio. And the idea here is basically we have a time going from the top to the bottom, and we have an environmental change here happening at this stage. And we have 4 time samplings here, time 1, 2, 3, and 4. And if we can detect some genes that have this trajectory, changing the allele frequency between time 2 and time 3, we could potentially associate that to an environmental pressure and relate this change, this allele frequency change, to this environmental change. And this has been done before. This work is from 2015, which to the best of my knowledge was the first work to do this. They were comparing mutations in those genes, the lactase gene, the Fas gene, which is omega-3 metabolism. There were some genes for pigmentation and other genes. He was comparing this allele frequencies in the past with allele frequencies from the present using ancient DNA and present-day data. And he sees this variation in allele frequency. Those genes, we know already that they are, in those variants, they are under the effect of natural selection. So we would expect to see this variation, and we see in this paper in 2015. More recently, there was this paper 2022 that used the same approach. There is Black Death occurring, and then you expect, if you look at allele frequencies for different genes that are involved in immunity, for example, before and after the Black Death, you would see this variation in allele frequency, like coming from low frequency to high frequency, for example. Those two studies, they have one thing in common, that they are focusing on genes that have mutations with a very strong effect And that effect, and those genes can be mapped to those mutations can be mapped to single genes. So this is a monogenic approach to study positive selection or local adaptation. This is good, but it's limited in the sense that we need very strong selective pressure to be able to see this change. So we were with this in mind, and that's what I mentioned before. Like those studies, they focus on large effect mutations that can map to single genes. With this in mind, and they miss signals that are natural selection signals, but that are mutations with very small effect, and also that act together with other genes in what we call polygenic architecture of some traits. And so we were thinking about addressing these issues, and my lab is currently doing, using the theory that we have for learning about natural selection with using single genes, the monogenic approach, and you're also using methods from human genomics that are studying polygenic traits to study polygenic selection using ancient DNA datasets. And the information from human genomics that we use comes from what we call GWAS, the Genome-Wide Association Study. GWASs are, it's much more complex than what I'm going to explain here. Here's just a brief idea. You have a large number of individuals, let's say 2,000 individuals. You sequence a bunch of different genes in those mutations in those individuals across the whole genome. And then you split those 2,000 individuals into two groups. Let's say one is carrier of a disease, for example, cardiovascular disease. The other ones are healthy, don't present that trait. And then you start to compare each variation that you find in the genomes of those individuals and see if there are genes for which a variant, an allele occurs more often in one group and not in the other. And by doing so, we can associate that variant to the trait of study. So that's what we call the genome-wide association study, in summary. And those GWASs, sometimes they show you maybe like the top 3-4 picks, which is what people discuss, but they actually give you an idea of this polygenicity of the traits, usually indicating that 100 or 1,000, few thousands of genes across the genome are contributing to a specific trait. And each gene actually contributes with very small effect, typically, but sometimes with larger effect. But what matters here is that there is a ton of variants, thousands of variants, and each one contributing with specific effect. Some will increase your chance to have a certain disease, or will prevent you to have that disease, and so on. You can add all those effects from all those variants and build what we call polygenic risk scores. For example, if we think about cardiovascular diseases and we do a GWAS here for this, for everyone here in this room, we could calculate a polygenic risk score for each one of us. I will have 2, you have 3, you have 1, whatever. There's going to be a distribution of these polygenic risk scores, and we can plot that for population. We can do that for this room, of course, but we can do for 2,000 individuals for whatever number of samples that we want, and build these distributions here. And some people will have low risk for the disease, some people will have a high risk for the disease, and some people will have, most people will have a medium risk, right? Most people will be like intermediate in the distribution, which is normal of most of the biological characteristics, right? So here's another, just to show another example here of what I mean with polygenic risk scores. We see the distribution of the polygenic risk scores there, and we see 2 individuals, one individual on the left, indicated by that green arrow. That individual carries 2 mutations that cause a disease that is being studied. The other individual on the other side has 7 of those mutations. So that individual there on the right has a higher risk to show the disease than the other one on the left. So this is the idea of polygenic risk scores. And we're going to be using those to, and apply those to ancient DNA data to understand the evolution of resistance and predisposition to infectious diseases. So that was not our idea, to get GWAS data from the UK Biobank and FinGen, for example, then build polygenic risk scores models to calculate each individual's risk for having an infectious disease or not, and then calculating that distribution of PRS, the polygenic risk scores, in different time points in history. This is the data that we have, that we used. At the time, the UK Biobank had 420,000 individuals of European ancestry, and the FinGen had 377,000. We were able to get 50 diseases across both data sets or traits that were related to predisposition or resistance against infectious diseases, and that also had at least 1,000 cases, 1,000 individuals in the original study that had a higher predisposition to infection, for example. And then we also want to see if the trait is actually heritable through DNA, right? So humans can inherit traits because they live with other people in their families, right? So even if you adopt a child and don't share a lot of your DNA with your parents, you are living in the same environment, and you're going to present a lot of traits that are similar, right? And also, so we want to filter those traits that are actually inherited through something else that it's not DNA, through the ones that are actually inherited through DNA. And we do that by doing this heritability analysis. So the UK Biobank and the FinGen have already the heritability of each trait, and we have a threshold to see what we accept and not as a heritable trait. So you can see out of the 50 traits or 49 traits that we analyzed, only 7 survived. Only 7 were heritable through DNA. And those are the traits. The names are a little bit complicated, and they don't matter very much. What matters here is that all of them are very broadly like bacterial diseases, intestinal infections. They are not very specific to one specific pathogen. We didn't find any of those that actually was heritable in these studies. So we picked those ones here, those 7. And the data set that we analyzed, we still haven't integrated our own data set there. We are using the data set called AADR, which I showed before, mentioned before. They have 10,000 human individuals, right? We focus on Western Eurasian population. We also use pseudohaploid data to control for that effect that I said, that excess of homozygotes. We, in human genetics, in human population genetics, we usually use this data set called the 1240K. Basically, it's 1.2 million SNPs across the genome that we sequence. We got the data for those 1.1 million autosomal variants. The other ones are not autosomal. And we selected the samples, the ancient DNA samples that had at least half of those variants. So 600 single nucleotide variation. The sample size that we get at the end is 2,400 individuals that are spread between 200 years ago up to 10,000 years before present. And we group them in intervals of 200 and 400 years, and we compare the results. They are always telling the same story. I'm going to show only the results for the 400 years intervals, and I'm running over time. So the idea here is that we have a polygenic risk score distribution before the outbreak that we can calculate. Then we know specific outbreaks that happen in Europe that is very well documented in the historical times. And we see if the PRS distribution remains the same, or if it's over-dispersed later, or if it's actually shifting towards higher risk or lower risk, and so on. So we do that for those 7 traits that I mentioned that are heritable. And we find that, that 4 of them actually show significant shifts in the PRS distribution across time. At this point, we already don't know which time it is varying. This first analysis is just showing like those many distributions, are they different from each other? And those four, the tests that we do for those four say yes. The PRS distributions are changing from one point to another. And I'm putting here some details. It's just we are correcting the significance for multiple tests. We are also considering different covariates. One that's very important is genetic ancestry. We know that in Europe there were several different migrations that affected the genetic ancestry of the individuals. We are accounting for that as well. And so what does this tell us, that PRS distribution is changing across time? It tells us that the predisposition to infectious disease is changing across time in Europe. And yeah, that's what it's telling. And what I'm going to show here is some of our results. I have the PRS distributions that I showed before like this, right? Like a normal curve. What I'm going to be showing now, it's a violin plot. Basically, I am shifting it to like 90 degrees and duplicating it. So it's going to be a violin. And that's what we see. I'm going to show the four that we have later. This one is the one that we see, visually we see the differences. So I'm going to show the differences here. So the first thing that we see is that the median PRS shifts across these times, right? So it's higher here and it's lower there. More or less following this blue curve. And the other one is that some periods are over dispersed, like the ones here towards that side of the plot. So I'm going to start with the present and go towards the past. So here are the two different traits that we were able to observe those differences in the FinGen dataset. And one is diarrhea and gastrointestinal infections. The other one is bacterial diseases. The other one for the UK Biobank follows the same idea, right? But are two different traits, basically bacterial infections. Here we don't see visually those changes that I showed before, but they are statistically significant. So what caused those shifts? We know it shifted, but when does it shift? We did another statistical test, now comparing each pair of those periods to see which ones were the top three in the case that have the most differences across those several pairwise comparisons. And by doing so, we were hoping to find the timing that this event that shifts the selection, the PRS distribution happened. And those are the three times that are the ones that have the most significant differences when compared to the other ones. One is from 600 to 1,000 years before present. The other one, 1,000 to 400 years before present. And then 1,400 to 1,800 years before present. So those are the time periods. And here I'm showing a figure from this review from 2019 from Maria Espiro. It shows historical and prehistorical known disease outbreaks that happen in Europe. And then we qualitatively, like matching with that, we sought to see when are those periods that we highlight as PRS changing fall here. And they happen to fall on those three different diseases. One is the Black Death, which is the most recent one. The other one is the Plague of Justinian. And the oldest one is the Antonine Plague. I have some information here about them. I'm gonna, for the interest of time, I'm gonna move a little bit faster. But basically, the Black Death is one of the, is the most fatal pandemic recorded in history. It killed 25 to 50% of the population of Western Eurasia. We also have the Justinian Plague that is thought to be caused by the same agent, the same bacterium that caused the Black Death. And then the last one is the Antonine Plague, which we think it's not the Yersinia pestis. It's still some debate what happened. And in the historical record, there are descriptions of the disease, but we are still unsure what happened there. So people think it's smallpox or measles. This is the very last thing that I'm going to show here. One thing that we were wondering is that if this selection, like let's say several genes, here's one gene that it's being selected by this interaction with the pathogens. We also see potentially some effects of this response to pathogens in other traits as well. There are different ways that we can do that. We went with a very simple approach, which is doing this gene ontology analysis. Basically, we got all the variants that are involved with a specific trait, and we see which are the pathways that are also associated with those variants. Beyond what we have already, that we know that it's immunity, right? So here are some of the pathways that we know that we would expect to be related to these genes, which is all of them that are related to immunity. But we also find other pathways that are not necessarily related to immunity that are also impacted by these events. One is the lipoprotein particle clearance, pigment granule localization, insulin stimulus response, and metabolic processes. So that's just a quick slide here showing that this process of a response to pathogen actually might have affected other pathways as well, other traits in humans. And here are some take-home messages in the last slide. This work shows that pathogens played a crucial role in recent human evolution, right? So this interaction between humans and pathogens significantly impacted our genome. The other thing is that we can use ancient DNA to learn not only which genes are under selection, but also when selection occurred and what's the strength of selection. I didn't talk about strengths of selection here, right? I talked about date, but we can also learn about strength of selection, as I mentioned before, briefly with those simulations. The other point here is that we were able to use ancient DNA to study the evolution of polygenic traits. So in addition to study monogenic selection, we can also use ancient DNA to study polygenic trait. And last, we see this involvement of this phenomenon of adaptation to host pathogens potentially impacting other pathways in humans as well. And with that, I will conclude and thank everyone who is here, in special Abby for the invitation. And thank everyone. And